As World War II began to wind down in the mid-1940s, the Princeton mathematician John von Neumann and the industrial researcher Vladimir Zworykin approached government officials in Washington, D.C. to request funding to create new computing infrastructure for weather prediction. They proposed a two-step plan. First, predict the weather. Second, control it. As described in their proposal, only with exact scientific weather knowledge will effective weather control be possible. A year later, a hurricane was forecast to remain at sea, and researchers thought it would make an excellent testbed for an attempt to dissipate one. They dropped 80 kilograms of dry ice from a B-17 bomber with the intent of disrupting the hurricane's internal structure. But the worst possible thing happened. The hurricane's trajectory changed 130 degrees, and it landed in Georgia. Welcome to Leafing Through Science, where we dive into the pages of popular books in the life sciences with the writers who made them happen, from new releases to timeless classics. I'm your host, Hedi Young, and today we're taking on one of the biggest challenges in medicine: understanding and treating the human brain. Alzheimer's, Parkinson's, depression, addiction, multiple sclerosis, schizophrenia, and many more. Almost every one of us has been touched, directly or indirectly, by brain disorders, and despite decades of progress in research, we still have no cures for many of these conditions. My guest today is Nicole Rust, a neuroscientist, professor, and author of the new book, Elusive Cures: Why Neuroscience Hasn't Solved Brain Disorders and How We Can Change That. Welcome to the show, Nicole.
Oh, thanks so much for having me, Hedi. It's a pleasure to be here. I appreciate it.
In Elusive Cures, Nicole makes a powerful case that treating brain disorders isn't like replacing a broken part in a machine. Instead, it's more like trying to redirect a hurricane. She argues that only by embracing the brain as a complex, adaptive system can we hope to make real progress. Nicole brings extensive experience at the front lines of neuroscience research. She's a professor in the Department of Psychology at the University of Pennsylvania, where she studies how the brain remembers the things we see and how it shapes the mysterious feeling we call mood. And alongside her research, she works to develop new therapies to treat memory and mood dysfunction. In this conversation, we'll talk about why so many brain drugs still come from discoveries made in the 1950s, before we knew much about the brain at all, why progress has been slower than many people hoped, and what new ideas and approaches might finally help us make breakthroughs. So, whether you're fascinated by the brain, touched personally by mental illness, or simply curious about how science evolves, this episode is for you. Now, the brain is often said to be the most complex object in the known universe. And that alone should already tell us we need to tread carefully. And you open your book, Nicole, with a story that really drives this home: Alzheimer's disease, which slowly erases memory. Carol Jennings noticed that her father and two of his siblings were all developing Alzheimer's unusually early, in their 50s. She had a feeling it must be genetic, so she started writing letters to researchers offering her family's help. And one of those letters reached Dr. John Hedi in London, whose team would go on to discover that a single DNA mutation, just one letter swapped in the genetic code, was driving the disease in her family. And at the time, this was hailed as a huge breakthrough. The thinking was, if we could clear out the sticky protein clumps called amyloid plaques that build up in the brain and are linked to this gene, we might finally have a cure. But fast forward more than 30 years since Carol wrote that letter, during which time Carol herself died from the disease, we still don't have a highly effective treatment or cure for Alzheimer's, not even for families like Carol's, where we can pinpoint the mutation. And in fact, in 2011, 6 of the world's largest pharma companies decided to pull back their brain drug development because after years of pouring billions into the venture, they still had little to show for it. And reports suggested that these companies had come to the same sobering conclusion. We don't yet know enough about how the brain works to create effective new treatments. And as you put it, by far the biggest factor holding back the development of new treatments is simple. We do not know enough about the causes of brain dysfunction.
Yeah, so as we dive into this, I just want to always begin by saying that when we use this term disorder and dysfunction and illness, we want to use that term a little bit carefully, not when we're talking about Alzheimer's. I think in the case of Alzheimer's, it's clear. Nobody's going to dispute. Alzheimer's is a disorder and it's a disease, and we want to cure it. But there are other types of disorders that we want to be a little bit more careful with. And so the concern is that some of these conditions that are labeled disorders, like attention deficit hyperactivity disorder, ADHD, and some forms of depression, they're actually forms of neurodiversity that society maybe has wrongly become intolerant to. And, the way I see it, and I think the way that many see it, is an individual has to decide for themselves with those conditions if they want and need treatment. And so when we enter into this conversation, we do so with a very empathetic spirit, both for Carol Jennings and her family, as well as anyone who's suffering from these conditions. The idea is that what we have to offer individuals right now really falls short of what they need, starting with Carol Jennings. So just zooming back out, when I set out to write Elusive Cures, as you said, I'm a brain researcher. I've been doing this for decades. And I am what's often called a foundational or basic researcher. So the idea behind the type of work I do and many other researchers like me is that we just don't know enough about how the healthy brain functions to try to understand what happens when it's dysfunctional. And so I like to liken it to mechanics. When we ask a mechanic to fix a car, it usually comes with some training about how the car works. They have a manual, they know how this car works. And our physicians right now, they're just dealing with these manuals. They're so full of these empty pages. We just don't even know how so much of our brains work because, as you said, the brain is really held up as the most complex thing in the entire known universe. So researchers like me are responsible for filling the pages of the manual, right? And the idea is that that knowledge is absolutely foundational. That is the bottleneck to understanding and treating brain disorders of all types, from Alzheimer's to depression. So when I set out to write this book, it was a personal journey for me to try to understand, why is it that brain researchers like myself have indisputably been knocking it out of the park for decades, right? In terms of the number of facts we've collected, the number of pages in our textbooks, they just keep getting thicker and thicker and thicker. Why have we been learning so much? But yet, these end goals, the things that society really needs for us, cures for Alzheimer's, better treatments for depression, why haven't those been more forthcoming? And there are cries for, we need to do something differently. And they're coming from the heads of agencies like the Wellcome Trust in the UK and the National Institutes of Mental Health in the United States. So for me, it was really trying to figure this out for myself as a researcher as much as anything. What is it going to take to get from where we are to those societal end goals? And the story of Carol Jennings really deeply moved me as a researcher who studies memory and thinks about that work as foundational for understanding and treating cognitive disorders of all types, including Alzheimer's disease. That story is a story of incredible human ingenuity, right? So there's no question what causes Alzheimer's disease in Carol Jennings' family. I do want to highlight that for most cases of Alzheimer's, it's not so clear. So I like to think about Alzheimer's in 3 kind of different categories. One, there are the very rare genetically inherited forms of the disorder, like Carol's family carries. If inherited, those will lead to Alzheimer's later in life with certainty. Then there are other genes that you can inherit, and they will increase the risk of Alzheimer's, but they will not ensure it. And then there are other cases where we don't have any sort of genetic links. It's just individuals get Alzheimer's and there's no clear genetic cause. But in Carol's family, there's no question what causes their disease. It is the mutation in a gene for a protein called amyloid. And we know, going back to Alzheimer himself, the brains of individuals with Alzheimer's, they tend to have clumps of this particular protein that's mutated in Carol's family. It's called amyloid plaques. So human ingenuity, researchers set out following that discovery. Carol, working with researchers, including John Hardy in London, to clear amyloid plaques from the brain. That is what's causing Alzheimer's disease in Carol's family. And it didn't work. As you said, it didn't work even in families like Carol's, where we know that's the cause. So what kind of system would do that? What kind of problem might be going on? And the new understanding is in families like Carol's, what's happening is amyloid is the culprit, but it's setting off a cascade of events that cannot be stopped by clearing amyloid from the brain. It's something much, much more complicated than that. And the idea behind many hypotheses about what's going wrong is we're working with a system that isn't, as you said, it's not just a broken link in a chain, a machine. This is a system where we have causes that lead to effects, but they're also feeding back on themselves as causes. So these are very complicated systems. We know a lot about these types of systems because there are other systems like that. They include things like the weather, ecosystems, nuclear reactors, and so on. And one thing we know is that small perturbations in these systems can have large and unpredictable effects. And that's probably what's going on with Alzheimer's. So Alzheimer's disease is not going to be as simple to cure as just clearing amyloid from the brain. We're going to need a different type of approach.
So to give people the lay of the land then, so for Alzheimer's and Huntington's disease, we still lack any highly effective treatments. Disorders like depression and schizophrenia, existing treatments work for some individuals, but not for many others. For the 8.5 million people with Parkinson's, we do have treatments that can help manage the symptoms, but there's still no way to actually slow down the brain degeneration that causes the disease. And to complicate matters further, many of these brain disorders might not be just one disease at all, but actually different conditions that we've lumped together under a single label. There can be a huge amount of variability in how these illnesses show up from person to person.
Mm-hmm. Yeah. So just for perspective, as we go into this, really well said. I don't want to make it sound like nothing has been happening because it has. We have had game-changing discoveries that have led to classes of game-changing drugs for certain neurological conditions. There are three classes of disorders, as you've mentioned, and so they are the neurodegenerative conditions like Alzheimer's and Parkinson's and multiple sclerosis, the neuropsychiatric conditions, including depression, anxiety, psychosis, and then the neurodevelopmental conditions, forms of intellectual disability and autism spectrum disorder. These are the three classes of disorders we've systematically been really struggling to figure out ways to understand and create new treatments for. It's not that we don't have any treatments for some of these disorders, and as we can talk about, some of them were kind of stumbled on serendipitously, but we really don't have a good understanding of what's causing them or what's happening in the brain. And so it's really those classes of disorders, the most formidable disorders, that I wanted to really focus on in Elusive Cures and trying to ask what's it going to take in order for us to understand and treat those.
And the problem, you argue, is that up to this point, the current grand plan of science, the molecular neuroscience framework, as you call it, has oversimplified how we think about the brain. Could you walk us through why this framework seemed so compelling and what you believe its limitations are?
Yeah, so rewind back to the 1990s, researchers were really excited about two transformative technologies: the ability to sequence genes. So, this was the era of the Human Genome Project, where we were able to do that and for the first time, and image brains non invasively. So, image the human brain non invasively through the skull using functional magnetic resonance imaging, fMRI. And there was just this general kind of optimism that if we just sequence genes and image brains, we'll figure out the causes of brain dysfunction and that will lead us to treatments. So if you just look at under the hood, like, why would they think that? In broad strokes, the way researchers were thinking about the brain at the time is a big domino chain, and it all starts with our genes. So our genes are used to create our brain cells. Our brain cells are wired together into these brain circuits. The activity of those brain circuits then gives rise to all of our functions as well as our dysfunctions when we have dysfunctional activity. And there was a single feedback loop in that whole idea where our environment feeds back to shape our brains, that's learning, and that happened via changing which genes were expressed, which then changed how the neurons function. So when researchers were thinking about the brain that way, there was immense focus on trying to understand different stages in the domino, right? So, how the expression of a particular gene would change how a brain cell functions, or how brain activity would give rise to behavior, things like thinking and talking and moving. And when researchers were thinking about causes of dysfunction, they were looking for the broken domino in the chain, which might be a mutated gene or a variant of a gene that might cause something like Alzheimer's, as we just talked about, or Huntington's disease, or it might be a brain area that has aberrant activity, which we can go in and recover via some form of brain stimulation. So that was the idea behind the approach. And the approach was successful in some cases, but in these other cases, it turned out not to be. So you can ask the question, well, why did we limit ourselves to thinking about the brain like that? After all, we've known all along that at all levels, there are these big feedback loops. So if brain area A sends information to brain area B sends information back again to A. Part of it is we just really didn't understand what those feedback loops were doing. And part of it was that we were technology limited. So 20 years ago, when I was recording from neurons, we could record from one or maybe two neurons at a time. We've now had these amazing breakthroughs. So we now can record from 1 million neurons simultaneously in a mouse. It's incredible. We also were very limited by our technology and how we could analyze that data. So at the time, we really couldn't deal with these models of the brain where you have feed forward and feedback connections. We could deal with toy versions of those, but we couldn't train a model like that to do something that could rival the complexity of human behavior, thoughts, movements, the ability even to recognize, the objects we're looking at. Today, we finally can do that for the first time. So it's the convergence of these two types of technologies, our ability to measure things we've never been able to measure before, and our ability to use new developments in AI and machine learning in order to deal with that data. This is allowing us to really tackle the complexity of the brain for the first time in history. And my speculation is that when we fast forward 30 years from now and we look back and we say, what was the genius of the 2020s? What was the big development in that era? We will discover that it was, in fact, this shift away from thinking about the brain as a domino chain and toward thinking about it as a system full of these feedback loops.
And yet, as you write, for a long time, the molecular neuroscience framework view has dominated how people in this field think and do their research. And so, to sum up, then, yes, the idea was that if we could pinpoint a gene mutation causing a disease, we could recreate it in a mouse, figure out exactly what was going wrong in the brain, and then design a drug to fix it. And one of the most powerful statements of frustration you write about comes from Thomas Insel, the former director of the National Institute of Mental Health. And he wrote, I spent 13 years really pushing on the neuroscience and genetics of mental disorders. And when I look back on that, I realized that while I think I succeeded at getting lots of really cool papers published by cool scientists at fairly large costs, I think 20 billion, I don't think we moved the needle in reducing suicide, reducing hospitalizations, improving recovery for the tens of millions of people who have mental illness. I hold myself accountable for that. And thinking of the brain as something where molecules were the key led to an intense focus on studying molecules. From 1990 to 2023, almost all the Nobel Prizes for brain research went to discoveries about molecules, like how we sense temperature and smell, how day and night rhythms work, and the molecular machinery inside neurons, such as ion channels and G proteins. By comparison, in those 3 decades, only one Nobel Prize was awarded for work related to brain activity and function, and that was for research on spatial navigation. But to be fair, this intense focus on molecules did lead to some major breakthroughs. For example, scientists discovered that neurotransmitters released in the brain get reabsorbed through a process called reuptake, which paved the way for antidepressants like Prozac, which works by blocking the reuptake of serotonin. Another big discovery was that people with Parkinson's have less dopamine in a part of the brain that controls movement, called the basal ganglia. And that insight led to the development of the treatment levodopa in 1970. And if you ask someone how new treatments are created, you're probably likely to hear some version of the bench to bedside story. The idea is that discoveries made at the research bench eventually lead to treatments at the bedside where they can actually help patients. And we'll get into why you think that bench to bedside treatments are actually the exception rather than the rule. But first, I think it's important to acknowledge that there have been some big successes. For example, could you walk us through the development of suvorexant? Not sure if I'm pronouncing that right, which followed from the discovery of a pathway in the brain called the orexin system to help people with insomnia.
Yeah, the development of suvorexant it's a fascinating story, and as you said, it's a wonderful example of this classic bench to bedside story where a breakthrough, a new discovery about the brain leads to a new therapy. So the discovery of suvorexant happened at a time in which researchers were aware that there are chemicals in our brain that help us go to sleep, like melatonin. What they didn't realize is that there are also chemicals in our brain that keep us awake. And orexin was discovered as the first chemical that does that. So it was discovered when they were looking at a version of dog narcolepsy. So there are dogs that suffer from narcolepsy. Like humans, they'll spontaneously fall asleep during the day. And in the dogs, unlike the humans, it's genetically inherited. So they knew there must be some sort of gene that's carrying it. Trying to figure out the gene that's responsible happened at a time before we had these really high throughput genetic sequencing techniques. So it took a very long time in order to identify the gene, but they found it, and it was an orexin receptor. They then were able to use that information to figure out what's happening in humans with narcolepsy, and they were able to make those links. In the case of humans, it's not genetically inherited, but they were able to figure out that what's happening is the brains that release this chemical in the brain, orexin, that keeps us awake, those neurons are actually degenerated and there's no longer any orexin. And so that's what's causing the human narcolepsy. And so for the first time, researchers realized, oh, there's this chemical in our brain that keeps us awake. And all that research, it took decades in order to get there. From there, the pharmaceutical company reasoned, well, maybe this is one of the problems why people can't sleep, why they have insomnia. Maybe they have too much of this orexin chemical, and so they can't go to sleep. And so, what we want to do is we want to block the receptor for this chemical to bind in order to trigger its stay awake effects. A remarkable story there in the pharmaceutical industry. So, a company called Merck screened 2 million chemicals to find one that would block the orexin receptors in the right way. They then even proceeded to take the chemical that they found and refine it to make it even better, so minimal side effects. They put it through clinical trials. The whole thing took about 10 years, about at the cost of around $1 billion just from the pharmaceutical industry side. And voila, suvorexant was created. So the whole story begins with decades of academic research to discover this new chemical in the brain that works in a way that no one ever anticipated and is involved in sleep. And then the pharmaceutical industry working for an additional decade to create this one drug that I understand for some people is very effective with their insomnia. So yeah, absolute epic discovery story from the bench to the bedside.
And now let's explore how brain drugs can come about in ways that don't fit the usual bench to bedside story at all. Take antipsychotics, for example. Not a single one of the 25 antipsychotic drugs, I was surprised to learn, reading your book, was developed through that classic pipeline of discovering a mechanism, designing a drug around it, and then testing it in patients. So, how were they actually created, Nicole?
Yeah, so the antipsychotics are a really interesting class of drugs. The first were developed back in the 1950s. It was actually a French pharmaceutical company who was developing antihistamines, right, for allergies, and they realized that some of their drugs had these sedative properties. So in 1952, they gave the first antipsychotic, chlorpromazine, to a patient that was manic and they had no other way to kind of alleviate the mania. And it was extremely effective, so effective that patient was able to leave the hospital sometime later. This then spread through institutions around the world, and it became a very effective drug to treat many, many patients with psychosis. It did have some side effects, but other drugs then followed. fast forward to today, all 25 drugs in this class of antipsychotics, they all work in the exact same way. They all bind to the same types of receptors in the brain, namely a particular type of dopamine receptor, D2 receptor. They actually bind to many different types of receptors, and their different side effect profiles are determined by the patterns or the strengths that they bind to these different receptors. It's not that none of the drugs in this class have been developed independent of any sort of information about the brain. In fact, when these drugs were refined to reduce their side effects, for example, the drug lumateperone was developed to reduce weight gain, it was developed based on extensive knowledge of how these drugs work. But the first drug was discovered somewhat serendipitously at a time before we understood much about the brain at all, just based on, oh, we're going to make some chemicals and then see how they work, see their effects in humans, and we'll try some stuff out. And then we figured out a little bit about how they work at the molecular level in terms of the receptors that they bind to. We're then able to refine them, but we haven't created a drug in this class that works in a new way. They all work in the same way as that original drug. So this is an example of not a new discovery in the brain leads to a new type of drug that works in a new way. This is an example of a serendipitous discovery a long time ago before we knew much about the brain at all. And then we refine those drugs based on information that we now know about the brain, but it's not a classic bench to bedside story.
Really good example of how the role of chance is an important factor in how treatments developed. I'd love to zoom in for a moment on schizophrenia because you use it as a powerful example of why molecular medicine and even measures of brain activity haven't yet given us the breakthroughs people hoped for, and what this tells us about the sheer complexity of brain disorders.
Yeah, the dream of the 1990s was that we would develop biological tests for psychiatric conditions, blood tests and brain scans, and that hasn't panned out. It hasn't panned out for any of the psychiatric conditions, including schizophrenia. So, yeah, there was hope that if we sequence genes, we can figure out the genetic causes of schizophrenia, and that would lead to insights. It's not that there aren't strong genetic links, it's just that those are very, very rare. And for most individuals, schizophrenia is weakly linked to hundreds of genes, not strongly linked to a single gene. For example, just to illustrate the genetic effects, if one identical twin has schizophrenia, the chances of the other identical twin having schizophrenia as well are only 50%. So that right there tells you this is not just about genes. It's also not just about the womb environment, right? Because those two identical twins were developed in the same womb. So there's something else that might be going on there. It must be some sort of environmental effect. It's also the case that brain scans haven't developed to the point where we can look down on a brain through a scan and we can diagnose schizophrenia in a particular way. We can do that neither with just looking at the structure of the brain nor what's happening in the brain. So even if someone's going through a psychotic episode, we can't even tell that by a brain scan. And there are lots of reasons that that might be true. Nobody really knows exactly why that's true. But yeah, blood tests, genetic tests aren't working out, brain scans aren't working out. It's not to say that we haven't identified anything. So one of the strongest links in schizophrenia, it's a over pruning of something called a dendritic spine. So when one neuron A talks to another neuron B, neuron B has these little, what they call dendrites. It's kind of the inputs. And those inputs have little things called spines on them that connect neuron A and neuron B together at the synapse. And there's an over pruning of those synapses in individuals with schizophrenia. That's a statistical test. We can't actually look at the brain of an individual with schizophrenia and predict whether or not they have the disorder or not. But it's given us some leads on what might cause the disorder. Today, it's considered to be a neurodevelopmental disorder. There's something that triggers this over pruning of these dendritic spines. We don't really understand what's causing that to happen. And like I said, there's no blood test or brain scan we can use thus far to diagnose it. So that's where we are with schizophrenia.
And now let's move on to how we can make inroads in our understanding of how the brain works. Back in the 1970s, researchers studying vision had discovered neurons that fired only when you looked at something specific, like a face or a hand. And that was an exciting finding, but as you write, scientists like David Marr realized this kind of observation wasn't the whole story. Just knowing what lights up in the brain doesn't actually explain how the brain builds a perception of a face or a hand, or why it works that way. And Marr proposed that to truly understand any brain function, you need to look at 3 levels.
Yes, and Mark called those why, what, and how, or at least I'll call them why, what, and how. They go by a number of different names. But yeah, and what was missing in the explanation you just described is the middle one, the what. So Marr really was keen into this question, what does it mean to even understand something in the brain? Like, what are we even looking for when we say we want to understand how the brain works? And when you have a neuron that is selective for a particular face or a hand, you know that the brain has solved it, but you haven't figured out how the brain has solved it. So Marr's top level is the problem that needs to be solved. That's the why level. One level down is the what level. This is kind of the hardest to wrap your head around. It's the part of I'll confess it's the part that I specialize in, the what level. But it's really something akin to a model or a mathematical equation that describes what the solution looks like. And then down at the bottom is how the brain implements that mathematical equation. And so often in brain research, especially when we're talking about drugs, we know a lot about the biological implementation. So things like a drug like Ritalin or methylphenidate, which is used to treat attention deficit hyperactivity disorder, we know how it changes how chemicals are released into the synapse. So we know things like it increases the level of that chemical. But there's a big question there. So how does that release of that chemical then translate up into the ability to pay attention better than if you didn't have that chemical in your brain? And that's that missing piece that Marr was focused on. It's that what level understanding. For the first time in history, we're starting to fill in that gap, and it's really exciting. And it includes discoveries about Ritalin. So, researchers recently have determined that this enhanced ability to pay attention, it's actually happening at the what level by decreasing noise in the brain and enhancing signals. And they can see how that's actually manifesting at the levels of individual neurons who are configuring their noise such that it averages out across these big populations of brain cells and brain activity. And that in turn, they can then link to how these chemicals are released into the synapse. So, for the first time in history, we have this multi level understanding. And I think that this is probably going to be an important part of the key to bringing back all the pharmaceutical industry into the game again, right? Because that was what they were complaining about. They were complaining, we don't know what causes brain dysfunction and we don't know how to develop treatments. We need leads to chase. And for the first time, we're starting to link information across all these different levels. How are things happening in the brain? What exactly is the brain doing it? And how is that leading to the why, the brain solution to treat brain dysfunction, like the inability to pay attention?
So, to sum up, then, Marr proposes that to really get to grips with a brain function, we need to know why the brain is solving a particular problem, what computations it's doing to solve it, the what, and how those computations are carried out in cells and circuits. And this can play out in different ways. You might get the same phenomenon having an effect across levels. For example, in Huntington's disease, a genetic mutation leads to a mutant protein that triggers a type of neurodegeneration and loss of function at higher levels. Or you might have many different types of causal influences interacting across levels, such as when genetic predisposition and poverty interact to cause addiction. And you describe this experiment regarding attention that you were just talking about, where participants have to keep their eyes fixed straight ahead while 2 streams of flashing bars appear, one on each side of their gaze. And they can't look directly at the bars. They have to use what's called covert attention to monitor them. And their job is to notice when one of the bars subtly changes its angle. When they get a cue about which side is more likely to change, they become much better at detecting it. And so this has allowed us to kind of make sense of attention in terms of these Marr levels, the what level increasing distinctiveness of stimuli, and the how level decorrelating noisy responses. And how does Ritalin affect performance in this task?
Yeah, so Ritalin enhances performance in anyone who performs this task, but it's particularly helpful for individuals who have ADHD. Yeah, so you will become more sensitive to detecting smaller changes. So anyone can see this. If I flash this and then I flash this, right? Everyone will get that. But if I flash this and then I flash something just very similar, you might miss it. And so you become more sensitive to detecting these small changes.
And this works for monkeys as well, I think,
And it
right?
works for monkeys as well. Yes,
Yeah.
when monkeys take Ritalin, the same effect holds.
So we have a pretty clear picture at all three levels then why Ritalin helps by amplifying attention, what it does, making the attended signal stand out more, and how reducing brain noise by enhancing feedback.
Mm-hmm.
But this multi-level understanding alone isn't enough. And so this is where we come to the next big idea in your book, which is that we need to start thinking about the brain as a complex adaptive system that computes. And you use the example of feedback loops, and we've already talked about feedback loops, such as how our bodies automatically regulate blood pressure, temperature, and blood sugar, and the brain is full of these loops. And
Mm-hmm.
why is seeing the brain as a self-regulating system so important for understanding mental and neurological disorders?
Yeah, this is a good question. And I'll admit, when I set out to write this book, I did not expect that this is where it would go. So for me, this was really a personal journey to ask myself, what's been holding back progress? And so I wrote through the chapter that we just discussed, thinking about things like Ritalin and filling in that what level understanding. And I thought that's the book I was writing. And I then realized like there's still this missing piece. So if we just back up, when we think about filling in the what level, it still is something like a domino chain of causes that lead to effects. And there's something else that happens in the brain. A brain's superpower, our brains, the brains of animals, all sorts of brains, the superpower of a brain is its ability to adapt to change, just like you said, to self regulate itself, whether it's temperature when you're getting hot and you begin to sweat, if it's blood pressure. So your brain is comparing the blood pressure from pressure centers in your body to what it should be and adjusting it, or if it's our ability to see a tiger and that triggering a whole host of chemicals like adrenaline and cortisol to just change our body's physiology to run and try to save ourselves, right? So we just have all of these different ways that we adapt to change, and that's one of the things our brain does. When you have systems like that, they most always exclusively require a feedback loop between some system that compares where you are with where you should be, what needs to happen, and then sending information back again in order to make that happen, whether it's sweating or changing your blood pressure or changing your physiology so you can run. So it creates these feedback loops. The issue is that adaptive systems, and it doesn't really matter if it's biological or if it's other forms like a power grid, these adaptive systems, they tend to be fragile. And so let's talk about the running from a tiger example. Those chemicals that are released in order to change our physiology, they are great when they're used just a little bit. But if you overuse them, they can actually burn a system out. This is what happens with chronic stress. So, one thing that can happen with chronic stress is it creates insomnia. Insomnia is the time of the day that our bodies tend to do a lot of healing of themselves. It can also create damage to certain structures of our brain, including a hippocampus, where our memories are stored, and there's a lot of other cognitive function. So this is happening because these systems, in order to be adaptive, which again is the superpower, it creates these interdependencies. So now you have a system that is dependent on sleep, and sleep is important for healing ourselves and so on, right? And so a lot of things can go awry in these adaptive systems because they are so fragile as well. And that's where so many of our disorders really come from. In fact, if you start thinking about the brain this way and the body this way, you can pretty much link it to every different type of brain and mental disorder. Our neuroimmune disorders are a great example of this. So, our immune systems have to do something exquisite and incredible, and that is seek out tiny little pathogens in our body and protect ourselves from them. So, that's a really fine tuned system. If that system becomes just a little bit out of whack and a little bit overactive, that's what causes an autoimmune attack. But we can't just shut off the system because then we'll die, right? We'll be invaded with different pathogens. So, our neuroimmune disorders, including things like multiple sclerosis, can be explained this way. Chronic stress can be explained this way. Other psychiatric conditions and neurological conditions as well. They can all be traced to this idea that what we really are dealing with here is not just a system that's a domino chain where causes lead to effects, but causes lead to effects that feed back as causes for adaptation, and that's what makes these systems fragile.
It's a really interesting example of how one's strength can actually also be one's weakness, because the same factors that lead to the body's adaptability also lead to its fragility. You point out that thinking about the brain this way as a complex adaptive system can help us understand the hedonic principle, the idea that we're constantly learning from what feels good or bad in order to approach or avoid things, and how mood itself is a kind of adaptive signal that shapes our learning and behavior. You talk about a fascinating study by Rutledge and colleagues in 2014 showing how mood can change depending on our expectations and experiences. Can you walk us through how this framework helps explain what mood is actually doing for us?
Yeah. So depression is very clearly the mood system kind of gone awry. And so, in order to understand depression, we arguably need to understand mood. Mood is interesting because, unlike other brain functions, the purpose of mood is not obvious, right? So, why do we see? We see to get information in the world into our heads. Why do we remember? Because we want to store information and use it later. Why do we move? It's because we want to pick stuff up and act on the world. But what is mood for? That's a lot less clear. As you mentioned, this general class of brain functions we call emotions are generally linked to this idea that they exist to motivate us to approach good things and avoid bad ones. Right. So they're a little bit unlike just putting a tag, a good or bad label on something. The way we can say, oh, I will call this a cup, right? That's just a label. This is a cup. But that doesn't make me want to do anything with the cup. I'm not motivated by the cup. The emotions, they take it a step above, right? Their motivation to approach good things and avoid bad ones. But moods are thought to be a little bit different than emotions, insofar as emotions are usually triggered by something. So if I see a tiger, I'm afraid. If I see feces, I'm disgusted. But we don't always know what's putting us in a good or a bad mood. So these moods are diffuse. What we would want from mood, to understand mood, arguably, is we would want a model of what mood depends on, right? Something that could say we experience things and this is how it impacts our moods. And this was the big breakthrough of the Rutledge experiment. So they came up with a reduced world. Let's start small. Let's even try to figure this out for a small world case. So the mood induction paradigm they used was a gambling task. So here's how it works. On each trial, you get to choose. Maybe you get to choose between winning 50 cents for sure, or you take a gamble where you might win $1 or lose and get nothing. Okay, so you have to choose between those two. We'll give you lots of different options of different types. Sometimes you'll win, sometimes you'll lose. And every few trials, they'll ask, how happy are you right now? So your happiness will fluctuate as you go through the task. And if your mood, your happiness depends on your wins and losses in this task, we should be able to build a model that says how happy is 50 cents going to get you, and how happy would be if you won it from a gamble versus if you got it for certain, and so on. And that's exactly what they were able to do. So what they found is that they could create a model for individual people in which they could perfectly predict how their happiness would fluctuate. Not perfectly, but actually quite good. I should say it wasn't perfect, but it was quite good. So what does it depend on? Well, it's not rocket science. It's like if you win, you get happy, and if you lose, then you become less so. But importantly, you don't have a really good access to what your mood depends on, what you're actually answering. And the reason, in part, is because you don't just care what happened on the last trial, you're averaging over several trials in order to create your happiness report. And so you can average, and then if you could take that little model, you can predict how happiness will fluctuate for individuals. So this is really exciting because it gives us some insight into what mood is all about. Part of the reason we don't have a really good sense of what's triggering our moods is because our moods don't just depend on one thing, like our emotions, right? I'm scared because there's a tiger there. I'm happy and I can't really tell you because my happiness is an average over many things. And from there, they were able to make some inferences about what then mood might be for, given that it's this integration across multiple experiences.
And it involves unexpected results.
Yes, yes. So if you win 50 cents and you didn't expect to win it because it was the outcome of a gamble, that will make you happier than if you won 50 cents and you picked it. So winning makes us happy, but surprising winning makes us happier even still. So it's not just about winning, it's about unexpected winning. It gives us a boost.
And so this is actually a different way of thinking about how the brain works, not as a information processing machine. I mean, it's difficult to understand what computing mood would really mean, but more as an organism that seeks pleasure and avoids pain, and mood being an integral part of helping it learn to adapt and find the pleasure and avoid the pain.
Yes, yes. So the idea is that mood exists as kind of an integrated average, a running average of goodness and badness that can help you actually learn. So in that example, classic reinforcement reward-based learning works by if you get a reward, you know that it's a good thing. And if you don't, you know it's a bad thing. And that's how we learn. We learn a lot based on that reward type of feedback. That type of learning works really well as long as the conditions in your world are very stable. But if the conditions are changing overall, and so imagine you're a monkey trying to figure out what trees have the best fruit in them, and it's spring and all of the trees are getting more fruit, or it's fall and all the trees are getting less fruit, you're going to confuse changes in the overall environment with changes in the particular items that are good and bad for you. And so the idea is that this running average helps us dissociate our individual experiences that are happening one by one to how things are happening for us overall so we can detect those changes and learn accordingly. That's the idea.
And of course, there are feedback loops here too, because mood affects learning and behavior, which in turn affects mood.
Yes. So now we have a system that is adaptive, right? It's for a good thing, it's to help us learn. But yes, our moods, and it turns, they affect our learning. They also affect our percepts. So we can experience the same thing. And if we're in a good mood, we'll perceive it to be better than it is. And if we're in a bad mood, we'll perceive it to be worse than it is. So now you have a system where our moods impact the percepts of the things that happen to us. And that is what mood is integrating over. So you have a system that, if you had too many bad things happen in a row, it could quickly spiral down into depression. Or a system, if too many good things happen in a row, it can quickly spiral up into a mania. And so obviously our mood systems have some sort of protective mechanisms to make sure that that's not happening too often. And those protective mechanisms are going awry. So yeah, just to step back, here's an example of a system where our adaptability to keep this running average is happening at the cost of this fragility where we can cycle down or cycle up. And that's really what it seems that depression is.
And that brings this episode to an end. But speaking of moods, hopefully it's left you in the mood for more. Because in part 2, you'll hear Nicole explain how protein folding won a Nobel Prize without anyone quite knowing how the problem was solved, how predicting salmon populations can help predict seizures in the brain, and how thinking of the brain as a complex adaptive system could offer a new grand plan for neuroscience. From building artificial retinas and uncovering why we sleep, to thinking of ourselves as processes rather than fixed things, and why the word cat might be better replaced with catting, stay tuned for part 2 and see you then.